Now it's time for our final graduate student oral presenter, Nicholas Ace Pugh. Ace is from Oklahoma City and got a bachelor's in science in biology from University of Central Oklahoma. After doing an internship with Dr. Dorothy Shippen at Texas A&M, he decided to apply here for his graduate career. He originally came from a molecular biology background, but he was exposed to plant breeding for the first time when he participated in Dr. Seth Murray's summer internship as a student worker before beginning his graduate career. He received his MS in plant breeding with Dr. William Rooney last year and is now a first-year Ph.D. student. His MS work was primarily concerned with determining whether or not sorghum could be improved for popping quality. One of his hopes for his career going forward is to meet the urgent need to produce improved crops that everyone can address the global climate change and world hunger. Everyone, please welcome Ace.
Thank you, Laura, for that nice introduction. As she said, I'm Ace Pugh, and my presentation today will be Heritability in QTL for Popping Characteristics in Sorghum Grain. So, why pop sorghum? Several companies already sell popped sorghum, and it's becoming increasingly popular with niche consumers. However, it is not the subject of the amount of selective breeding that popcorn has received, and there are few releases of improved lines for popping. So the objectives for this study were to determine the heritability of popping quality traits in sorghum, to determine the relative effects of genotype, environment, and genotype by environment effects for popping quality, and to identify QTL markers for popping quality. Now for this study, I used a population of 130 recombinant inbred lines derived from a cross between Serranio and RTX 430. And Serranio had a white endosperm and small kernel size, and it also has a relatively good popping quality for sorghum. And TX 430 has a yellow endosperm, a large kernel size, and a poor popping quality comparatively. And this was grown in a randomized complete block in the field with 2 repetitions in 3 environments in Texas. So next I'll discuss briefly how I phenotyped the 2 traits of interest of popping efficiency and expansion ratio. So first, heated air poppers for popcorn were modified with a wire mesh because the sorghum grains were actually so small that they would be ejected from the machine if that wasn't there to stop them. And I counted samples of seed for each recombinant inbred line. And the pre-popping volume was taken in milliliters of these seed samples. Next, the samples were heated for 2 minutes and 15 seconds, and 2 hot air poppers were blocked with each field rep. The popped samples were poured on a 4.7 millimeter sieve as shown, and the kernels that fell through the sieve were considered unpopped. Finally, the volume in milliliters of the popped material was taken, the unpopped material was recounted, and these measurements were used to calculate the popping efficiency and the expansion ratio. And the equations for these 2 traits are to the bottom right and left. The popping efficiency is essentially a measure of what proportion of the kernels actually popped, and the expansion ratio is basically how much did the popped kernels expand when they did pop. So what was the heritability of these popping characteristics? To find this out, a combined analysis across locations was conducted, and broad sense heritability was calculated on an entry mean basis using the equation shown. The statistical model for this combined analysis was fairly typical with the addition of the popper variable, and it was nested within the rep by environment as shown. All effects in this model were considered random. And this is the ANOVA for this data across all 3 locations. And what is shown here is basically that the effect of the environment, the genotype, and the genotype by environment are all significant for these traits. Now, interestingly, the environmental effect for these traits is probably the highest. But it is still clear that the genotype as well as the genotype's interaction with those individual environments is extremely important. And so the heritability estimates shown here kind of corroborate that by showing that the traits can be improved. Now the heritability for height is also shown sort of as a reference. Now obviously these heritability estimates are just within this population, but it wouldn't be unreasonable to presume that the trait is probably heritable in other situations as well. So in conclusion, for that part of the study, popping quality traits are heritable within this population, and improvement of popping traits in sorghum via selective breeding is likely possible. Next, I'll discuss the quantitative trait loci. Genotyping for this study was conducted by the Klein Laboratory using the digital genotyping methodology, and a genetic linkage map was constructed using JoinMap 4.0 software. And then QTL cartographer was used to conduct the QTL analysis. And shown here is the linkage map used for this study. Now this population had actually been previously mapped in a 2001 grain mold study, but that map clearly had a lot less markers and a lot less length, and the density was lower. This study had 828 markers spread across 1,527 centimorgans. QTL for popping efficiency were identified, 5 in total, and these were not seen in multiple environments. So basically each of these QTL was unique within the environments in which they were found. This kind of goes with what I said earlier, where the environment is heavily important for these traits. And I think it's worth knowing that out of the environments I did study, halfway was by far the best for popping. Now also here you can see some QTL that were found for expansion ratio. It's interesting to note that the QTL for expansion ratio, the alleles all came from the Sorghum parent, whereas for popping efficiency it appears that both parents contribute to this trait. So in summation, the QTL for PE and ER were not consistent across multiple environments. As expected, QTL for expansion ratio were all associated with the Sorghum allele. The lack of consistency of these QTL indicates that the traits are complex, just as they are in popcorn. So in closing, I would like to just thank everyone that has helped me on this study. It wasn't done solo, and I just want to say thank you to everyone that worked on this. And thank you to the committee of graduate students that organized this event today also. And with that, I'll take any questions.
All right. Thank you, Ace, for your presentation. We have just a couple minutes for questions. If you have any questions for Ace, please raise your hand and we'll bring the microphone to you.
Okay, I'll go first. So my question is about the demand for popped sorghum. I didn't even realize this was something that was available as a snack food or anything else. So it sounds intriguing.
Well, like I said, it's kind of right now it's a very niche market in the United States, but in other parts of the world it's actually a pretty common snack food. So I would just say it's a growing market. Let's put it that way.
Go ahead. A couple of questions. Are there any structural correlates that you've been able to detect in terms of the actual physical attributes of the seed? And secondly, do you have any idea as to why halfway seems to be a better environment?
The extra slides. Oh, okay, that's it. Thanks. Oh. This one. So yeah, I actually did not include that in the main talk, but I did run some correlation analysis on a few other traits to try to see if they also influence popping in any way. And I didn't find any very strong correlations, but I did find a lot of significant correlations between these traits and popping quality. And your second question was, why do I think halfway is the best environment? I'm not entirely sure, but I would hypothesize that's because it's a drier environment. So that probably produced grain maybe with lower moisture content, more suitable to popping. But that's just a hypothesis.
Is production of popcorn kind of a geographically localized industry?
Yes, yeah.
All right, thank you, Ace. It was a great presentation.